Professor Carafit coming to you from the biology bunker for Bio 1441, the clandestine online version here. I'm going to add in some information from the nervous system that was not originally in the syllabus, but I think it is important to cover. So this is chapter 48. And rather than assign you readings, you're welcome to read the chapter. I think it's important if you're going to med school and things. But at least review the chapter review. That's on page 1081 of your book. Read through that, and then also I would recommend that at least browsing the chapter and looking at the figures and reading the captions and things. I want to point out, in case you've forgotten, since you're all at home, at the end of each chapter in your book, there is a link. I know it's hard to see from a distance, but there's a link that you can type in or scan to practice tests for each chapter. Okay, and there's also a link at the beginning of each chapter to vocabulary quizzes, so like flashcards for vocab for the chapter. And that's what I recommend you do to help not fall behind on your studies. It's going to be real easy to accidentally fail a course if you don't keep up at home. So I understand that. Now, where we're starting today on this lecture is this critter. This is one of the coolest little critters on Earth. It is a cephalopod. You can see that it's got these blue rings all over it. It's called the blue-ringed octopus. And it's real little, not very big, but it will kill you dead. Not very big. You can see how small it is there. It's got a small bite. It's got some toxins in its saliva. It's got a beak just like the squid. Bites you, you get a little bit of saliva in your blood, but it has a neurotoxin, and that neurotoxin blocks sodium channels. So if you remember cells, like any of your body cells, you know, some of them have like sodium potassium pumps and things, and some of them have channels in their cell membranes that let sodium through. I'm trying to write as well as I can here. They let sodium through, and what this toxin does is it is able to get in there and plug up these sodium channels. Just plug them up, and it prevents sodium from flowing. The question here that's under the picture is that really a problem? Like, so what? Some sodium doesn't cross a membrane. Whoop-de-doo! What's that gonna do? Well, it's gonna cause you to not be able to breathe, and then you're gonna die. It's sort of important. So let's start with the basic layout of nerves. These things we call nerves, which are these basically like a tissue. Nervous tissue is made up primarily of cells like this. These are neurons, and they have a couple of major parts. We're not going to learn all these parts, but they have the cell body. That's the major part here. That's where the nucleus is and all the organelles you're familiar with. Okay, then they have this long part called an axon, and they have these little branches off the cell body called dendrites. Okay, so draw to learn, as they say. If you don't draw it, it's harder to remember. So you have a cell body, just like any other cell with your nucleus and stuff. It's hard to do that anyway. All right, and we have dendrites. You can label these on your own drawing. And then you have the axon. And then at the end of the axon are these little branches. So these are the synaptic terminals. That's important. Axon is important. And cell body and the nucleus, you know, all that, and dendrites. So those are the important parts that we need to know for now. I can't draw worth a crap with this thing. All right. So what happens is signals come into the cell through the dendrites. They're called dendrites because dendrology is the study of trees. Dendrites look like trees. Signals come in, travel through the axon to the synaptic terminals, and then to the next cell. Okay? And the way that signals are sent from one cell to the next is not an electrical impulse, but chemicals called neurotransmitters. Neurotransmitters come out of one synapse and into the dendrites of the next cell. Or if you're talking about, we talked about signals in the earlier lecture where the nervous system actually releases neurotransmitters that act as a hormone. So just as a reminder, hormones I didn't define this terribly well in the last lecture, but hormones, by definition, are chemical signals that get carried in the blood all over the body. Whereas like an autocrine signal is just sort of a self signal, you know, where it signals itself. Anyway, so by definition, a hormone's going to travel into the blood somewhere. So if you have a neuroendocrine signal, that neurotransmitter might go into the blood and go around, or it may affect the body in some way like that. It might stimulate a gland or whatever. Now, neurons typically are at rest, and they call this resting potential. This weird word potential has to do with electricity, okay? They have resting potential. So a neuron at rest typically has a negative charge inside, and the interstitial fluid outside has a positive charge. I'm going to grab my coffee. All right, so we have a negative charge inside and a positive charge outside. That's resting potential. And they maintain that with the thing you learned about in Bio 1 you can review called the sodium-potassium pump. So 3 sodiums are pumped out, and for every 2 potassiums pumped in. And that might not make sense, but when you have 3 positive outside and 2 inside, when you have that ratio, that means the inside's negative in relation to the outside. Okay? And also there are negative proteins in the cell that help to make it more negative. So we have a cell that's negative inside, positive outside. And then we have these potassium channels that let some diffusion take place as well. So we're pumping, we're diffusing, so some of the sodium leaks out, and I'm sorry, some of the potassium leaks out, I should say, of the potassium channels, and that also helps to maintain a negative charge inside and a positive charge outside. Lots of YouTube videos on this if you want to see it. All right, that means that we have potential energy here because we have negative inside, positive outside. We have things that want to diffuse across the membrane. That is potential energy. Just like when we make ATP, we have ions floating across membranes helping to make ATP. We have a potential to generate energy here. So here we have a neuron, a lot going on here, but we have sodium, potassium pump, we have potassium channels, sodium channels, and this is just a neuron hanging out. The inside of the cell down here is negatively charged. Let me turn on the fake laser pointer here. Negatively charged down here. Up here it's positively charged. Sodium potassium pump is pumping 3 sodiums out for every 2 potassiums in. So we have a negative charge in the cell, positive charge in the interstitial fluid out here. And there are some channels open allowing some things to diffuse back and forth. I'm not going to worry too much about that. Now that's called resting potential. That's when your nerves are not firing a signal. All right, but what happens? So here's resting potential. Resting potential, the inside of the cell has a negative charge, right? Something like negative 70 millivolts or something, okay? But what if you receive a signal, like a fly lands on your face? All right, now what? Well, that's going to depolarize the cell a little bit. I'll talk more about that. You might not feel it though if it's not enough of a signal. But if that signal crosses this magic line, which is like negative 65 or so millivolts, you don't have to memorize that number for me. But if you cross this thing called the threshold, then the nerve fires, and that's what this is showing. And that creates what we call an action potential. So if there's a stimulus hard enough, you're going to trigger a change in the cell. And look what happens inside the cell. The cell goes from a negative like 70 millivolt charge to a positive charge almost instantly. This is in milliseconds down here. And then it's going to return back. It's actually going to go lower in negative charge and then return back to normal resting potential very rapidly also. So we go from resting potential to a little bit of a signal to boom, firing. The inside of the cell becomes positive, and then it returns back to resting potential. So here's how this is going to work. 1, resting state. Normal things going on. Your sodium channels are mostly closed. Most of your potassium channels are closed. And we have the sodium potassium pump running. The outside of the cell is positive. Inside of the cell, the cytoplasm is negative. Number 2, you receive a little bit of a signal. We can see that here. A little bit of a signal. What happens when there's a little bit of a signal? Like something walking on your hand or whatever? That causes some of these sodium channels perhaps to open, allowing sodium to flow in. Some of these channels they call them pressure-gated ion channels. When they are pressurized, they open and they allow diffusion to take place. So ions diffuse in because of that channel being open. If it's a strong enough signal, lots of ions open. Okay? So if there's a lot of signal, a bunch of them open, and then we reach this phase here they call the rising phase of the action potential. I'm not going to test you on all of these stages. I'm going to ask you about resting potential and action potential, okay? And how we get back to resting potential. That's what I'm going to ask you. So, rising phase of the action potential. Why is it rising? More channels open, okay? That means lots of changes in charge. So notice, these charges out here are positive. They're flowing into the cell. They're making the inside of the cell positive. All right, so that's how a chemical signal is made. So why our electrical signal is made? We're changing the charge of the cell, like we're flipping it, and that's how the electrical signal is sent down your nerves. Then we close up all the ion channels, and then what we're going to do is fire up the sodium-potassium pump and things. And we're actually going to let potassium out of the cell to help bring the charge down. Then the sodium-potassium pump's going to bring us back up to normal levels. So here's what happens inside of that neuron. So you receive a signal. It opens sodium channels. So step 1, sodium channels open. You can write that down if you want. Step 1, sodium channels open. That causes sodium to rush in. That makes the outside of the cell more negative in relation to the inside of the cell, but just in one spot, right? So think of this as in the axon. The axon's just negative in one spot. The rest of it's still normal. But when this part of the cell becomes positive, that causes sodium channels to open down here. Those are voltage-gated ion channels. They open up when the voltage changes. And so that allows sodium to enter in here. Meanwhile, back here, we release potassium that stops the signal, so it reverses the charge again back to where it used to be. Then the sodium-potassium pump kicks in and brings it back to normal. So we stop this signal back here by letting potassium out. Then we bring everything back to normal down here by using the sodium-potassium pump. So we let sodium out, but remember, that's not where we want to be. Where we want to be is sodium in, I'm sorry, I'm messing that up. Where we want to be is potassium in and sodium out. So sodium is inside right here. We let potassium out just to stop the signal, to get it to become more positive outside than inside. Then the sodium-potassium pump balances everything out where it needs to be. So that's what's happening in the diagram. So if I were going to try to write on that, let me see if I can get this to work. Okay, so here we have 1 sodium Na plus channels open. Okay, sodium flows in. Notice the charge is reversed, right? Positive inside in this area. 2, potassium is let out. Their channels open. That makes the outside positive again. But it's not where we want to be. If you remember what resting potential was, it was lots of sodium out, lots of potassium in. So 3, the sodium Na plus potassium pump restores resting potential. Okay. Meanwhile, more channels open down here, letting the signal send down the neuron. I think we'll just skip this part since we're not meeting in person. I'll just let you know what this is showing. Your neurons actually has like insulation on it called myelin sheaths, and that's what this is showing, but we'll move on. At the end of the neuron, when the electrical signal caused by the change in polarity reaches the end, that causes exocytosis of chemical signals, which are sent from one cell to the next, and then when the next neuron receives that signal, it might open up its channels and allow sodium to diffuse in, and the signal continues. And that's how a nerve signal is sent from like your brain to your toe, for example. Now, I wish this didn't move over my words, but that's okay. So, blue ring octopus bites you. It releases a neurotoxin that blocks sodium channels, and then what happens is paralysis sets in, your diaphragm becomes paralyzed, and you can no longer breathe. Why does your diaphragm become paralyzed? Well, when your diaphragm is paralyzed, or when sodium channels are blocked, you receive a signal, like your brain says, breathe now, but you can't, because you can't open sodium channels and cause the charge of your neuron to change. It just stays in resting potential, and you're stuck. Meanwhile, the poison dart frog is well, it doesn't make its own poison, but it eats beetles and concentrates their poison. It produces a toxin, or has a toxin, that causes sodium channels to become stuck open. So then you're stuck in action potential. You can't restore resting potential. So the important things to note from this mini lecture here is that osmosis and active transport, I shouldn't say osmosis, diffusion and active transport are vital to understand how physiology works. The way you think, this system, this simple system of diffusion, is how you think, how you are who you are, how you remember things, and how you act. Without it, without simple diffusion, you're a lump of non-thinking material. Okay? Very important to understand that. So I'm going to leave it at that level. Okay? So stimulus happens. Sodium diffuses in. That causes more sodium channels down here to open, because the inside of the cell is now positive. That causes these channels to open. That causes more sodium to diffuse in, and that changes the polarity of the nerve. That's an electrical signal. Then potassium's let out. That stops the signal. And then the sodium-potassium pump returns things where they should be, where you have 3 sodiums out to every 2 potassiums in. That's how nerves work. And nervous systems are found throughout the animal kingdom, from Hydra and the cnidarians. Platyhelminthes have a little bit more complex nervous system. So these cnidarians have a nerve net, which just means like nerves are just spread out throughout the body. As we move up to like Platyhelminthes, they have eye spots, and they have an actual brain, or something like a brain, where nerves are clustered in the anterior end. And as we move up to vertebrates, we definitely have a large brain in the anterior end near the head, where the major sense organs of sight and hearing are located. Anyway, that's the nervous system in a nutshell, and we're going to move on to plants now.